Today, our speaker is Dejan Marković. He did his PhD in electrical engineering at UC Berkeley. And then he told me today that you actually went and did some private industry work for a short period of time.
That was
right after
work, yeah.
Sabbatical work, okay. And then he's been a professor of electrical engineering at UCLA for almost 15 years, I think you said. And he's done some amazing work in the design of neurostimulators, neurorecording devices, primarily work, I think, at least that I'm familiar with, as part of the DARPA program. So he played a huge role in the RAM project, and then more recently in the SUBNETS project. And I think that's what we'll hear at least some about today. And thankfully we were able to schedule his visit at the, probably the last nice day of fall in Cleveland. So he didn't have to be subjected to any brutal winter conditions. And he got to escape the fires that actually closed UCLA yesterday, he told me. So without further ado, let's hear what he's got to say. Thanks. Oh, here is a gift from the Cleveland FES Center. I'm supposed to give this to you at the end, but I forgot and brought it up now, so you can deal with that.
All right, thank you.
All right.
Well, thank you, Cameron, for the introduction. Thank you, everybody, for having me here. I guess I'm lucky to be here, given the weather and other circumstances. So I'm trained as an electrical engineer, but after I joined UCLA, I began to heavily slide into a new opportunity for technology in medical devices, which I find very fascinating. So today I will talk about closed-loop neuromodulation from the technology perspective, addressing the large unmet need for clinical purposes. So there is a large amount of accumulating clinical evidence of the efficacy of closed-loop deep brain stimulation, or DBS. There are two studies of the Parkinson's disease, one in a 4-patient intraoperative study that has shown that if you trigger adaptive stimulation on the power of the beta rhythm, that with 45% of time of stimulation as compared to the open loop, you achieve comparable therapeutic outcome. There was another 2-patient study with Activa PCNS conducted by Dr. Phil Starr and colleagues at UCSF that has shown similar effect, that if you, again, run the stimulation loop triggered by the beta power, you can actually save 40% of energy, meaning you put less energy into the body to achieve therapeutic outcome. There is also a nice article by Dr. Lozano published last year that talks about the need and opportunity for closed loop, particularly for cognition that requires phase-locked stimulation to be able to decode and encode memories. And there is also a recent article of last year from colleagues from the UPenn who have conducted a large patient study, over 200 patients under the DARPA RAM program, to show the effects of stimulation of closed loop for active memory restoration. So in order to address the needs of closed loop, which are limited by the ability of today's technology, there are several technology gaps to identify to be able to provide through closed loop technology. First one is we need the ability to sense from a large number of channels to achieve patient-specific localization. Second, we need sensing that is concurrent with stimulation, so that you don't blank recording during stimulation if you want to talk about true closed loop. Third, when you have a large number of channels, you want to be able to have real-time wireless data telemetry at rates that are far beyond Bluetooth low energy capability. I'm talking about megabits per second and more. And also, with higher number of contacts and all these sophistications, you need at the same time to miniaturize technology to be less invasive and more appealing to the patient. So I'll be talking about technology aspects that address those needs, and with understanding that technology modifications in medical devices don't happen very often, so when you have a chance to do it, you really kind of need to look carefully across the board what's needed and develop new platforms. And I would like to also give a little bit of a plug for UCLA. Today we are actually celebrating 50th anniversary of the internet at UCLA. There is a big event going on campus to celebrate that. But I would like to pay tribute here to another technology that was developed at UCLA, which is the brain-computer interface. So the term BCI was coined at UCLA by Professor Jacques Vidal in 1973, and in 1976 he has shown the first experiment in which it was possible to control a cursor in a two-dimensional maze through volitional brain control. And what's interesting, if you look into this paper from 1973, you see kind of three sections. On the left, there is the experiment room with the displays and EEG sensor that is mounted on a subject that is moving a joystick. Then in the middle, there is a control area with the computer algorithms that take this collected data, EEG recordings, and close the loop. And then there is the computer area which perform these computations. Back in those days, we had computers that had as much as 4 kilobytes of memory. And this first computer, actually, the node here that is shown is connected to the ARPANET, which is the first node of the internet. So this is kind of combining two revolutionary technologies of UCLA, the internet and the brain-computer interfaces. So from the 1973 until today, the very concept of BCI has not changed very much. And so we need to appreciate and understand those time horizons when it comes to bringing new technologies to clinical environment. So just to give you a bit of perspective here, in 2000, which was about 25 years later, this was a famous discovery from the Duke University where they have shown that you can transfer brain waves from a monkey at Duke through the internet to MIT lab to move a robotic arm. So I like to say this is putting two UCLA technologies to great use, which is the BCI and the internet, to enable these kinds of outcomes. Several years later, in 2003, this was an experiment in which monkeys were doing navigation tasks, and they were first moving the joystick to the point that they realized they can achieve the same outcome with brain control, and realized that we can just control the 2D navigation with mind. And then there is a translation to human patients in 2017 by colleagues from Stanford University who have published a study showing that a patient who is paralyzed can type on a computer keyboard, again using the same concept of 2D navigation from the founding BCI days. Now, this is kind of the neural interface technology applied to the human patients, that is the Utah electrode array that is then connected to an external device which collects all this data and then sends it further downstream to a computer for decoding and navigation tasks. So the BCI originated in 1973, but it was the first in human demonstration in 2002. The most recent BCI technology was published in July 2019 in a white paper by Neuralink, a startup in the San Francisco area which has developed an impressive robot tool that is shown on the far left. It has the capability to pick a large number of hair-thin electrodes and inject them with a couple 100-micron distance from each other in a very dense pattern of a few millimeter diameter. This is typically done for cortical interface, for motion and BCI type of experiments. And they advertise a couple thousand channels, so it'd be interesting to see what kind of learning we get from these additional channels for these kinds of application, and how and to what extent is this going to translate to actual human clinical practice. It is also interesting to see the kind of equipment that is used to do clinical work, because when we work with neurotechnology, one thing to keep in mind is that as much as technology sounds interesting, it is really irrelevant unless it really benefits a patient and clinician. So I like to always remind people that everything starts and ends in a clinic. We have to carefully look into what are the limitations of existing technologies and to what extent we can provide patient and clinician benefit. And so this is the clinical setup for science of human memory, which is actually afforded by the intracranial epilepsy monitoring for patients who are resistant to medication. They're implanted bilaterally on both sides, and they're admitted to hospital for 2 to 3 weeks to monitor the progression of their epilepsy to the extent to which the seizure locations can be localized, and then after the leads are explanted, subjected to curvature resection, or if that doesn't work, go through a second stage surgery to implant another device for therapeutic reasons. But in terms of technology, these are wall-plugged instruments that have typically something like a whole bunch of amplifiers and a computer and a screen that you can move around. The probes that are used on the far lower left, these are the probes we use at UCLA, which are used over 2,000 times so far. It's essentially LFP probe with a whisk of microelectrodes that is coming out on the most distal end to be able to record single unit activity for studies of human memory. And so it is with this justification of epilepsy monitoring that scientists get a chance to study things like memory areas. That only through the overlap of these areas that happen to be in the medial temporal lobe, in this case, we can actually study the science of human memory during situations when patients are not seizing and are able to perform the experiments. And so these are some of the examples from famous studies by Dr. Fried at UCLA, who is famous for his single neuron studies, that it was established that unlike motor cortex in BCI, studies of memory are very different because memories are non-topographic networks, meaning the two adjacent cells can encode entirely different concepts and ideas. And so it was found that, for example, in this case, one cell decodes strawberry, another cell next to it decodes, for example, a fish and a sailboat, which is also kind of giving rise to the theory of associative learning, that you can associate and create concepts. And so this is a movie from one of the experiments in which the neuron in the medial temporal lobe in the gray matter is responding to various stimulus that is coming from just like movie clips of people interacting in various environments, and this neuron is completely quiet except when Jerry Seinfeld shows up. And you can see that neuron is really responding to Jerry Seinfeld and pretty much nothing else. But it was also discovered that this same neuron responds to other characters from the Seinfeld TV show, which is, again, explaining the associative learning part. And it goes on with the association to Seinfeld's up to the point where there is no more Seinfeld content. And so this is highly selective, which also puts kind of the requirement that in order to really decode specific memories better, which are in a large non-topographic, non-overlapping, partially overlapping networks, you need many more channels to be able to have this capability. And longevity of really single units hasn't been established due to the lack of interface and the opportunity to do this in a chronic fashion over a clinically viable period of time. Now, the technology that is currently used for DBS is well established. So from the date it originated in 1952 by Delgado to the FDA approval in 2003, a long time has passed. And this technology has reached its maturity point, being out there for more than 15 years, where you have simply open loop system that is active 24/7 at 130 hertz, where clinician has only one knob, which is the amplitude of the stimulation current to control. And I'm sure this has been done, especially here, on a huge number of patients. UCLA took the opportunity to kind of promote milestone surgery, which was surgery number 500 in the social media, to increase the public awareness of this procedure that was performed by Dr. Nader Pouratian. And the patient happens to be a famous musician and actor, Brad Carter, who was playing a guitar during this live brain surgery to properly
Actually, it is brain surgery.
localize the
UCLA
position
implanting a
of the lead.
pacemaker
You can
in
see
Brad
that.
Carter's brain to treat hand tremors. But check this out. During the operation, the actor and musician was awake and he was playing the guitar. Strumming the chords actually helped guide the surgeons as they placed an electrode in his brain.
My music's really important to me, so I'm hoping that this will allow me to do that, to play guitar. I'm a finger picker. I want to record. I want to perform live again.
It's the 500th time the UCLA surgical team has performed this procedure. It's the first time that they tweeted it and had a live stream.
That is so cool.
Isn't it?
So that was the technology for Parkinson's disease and movement disorders, which has proven to be efficacious. And in some sense, the early success with the Parkinson's disease has sidetracked the field to the point that people were trying optimistically or naively, in a naively optimistic way, to use the same technology to other indications which are far more complex, such as depression trials, Alzheimer's trials, which are ongoing. And the results weren't as encouraging as in the Parkinson's. And this comes to really the lack of technology to properly access these indications and be able to even diagnose before doing the treatment of these kinds of conditions. Because DBS technology is limited to 4 to 8 contacts, which is really recognizing the brain world at the level of continents, where in some indications that are more complex, we really need much more intervention ability. And you can't access brain networks with such limiting resolution. We have limited or no data capability. Nevertheless, it is very efficacious in Parkinson's disease. There is another device which is cranially mounted from Neuropace, which is approved for the treatment of epilepsy, which has limited utility so far with 300 patients per year. And if you look into the social implication of chronic pain, Alzheimer's, and depression, we see that this is amounting to over $1 trillion spending per year to manage these indications. And what's common in them, those are network scaleopathies, where you require to access functional networks of the human brain and intervene accordingly. And epilepsy monitoring, in which you see that you can have multiple locations in the brain, is really a premium opportunity and a gateway to access these indications for translational science purposes and be able to inform future technology development. Now, before we go into what kind of technology is needed, it would be useful to reflect on the current status of neurotechnology. So we have, essentially, the conclusion is we have limited technology, meaning limited therapy, with the exception for Parkinson's disease and Cochlear. Those two things are efficacious because the tools are at the right scale. You have a multi-channel Cochlear interface with 65,000 implants per year, about 600,000 to date since 1985, and we have a Parkinson's at about 10,000 per year. All other indications are far below the need. As you can see, these numbers represent the number of people worldwide with certain indications. If you just take depression and Alzheimer's, that is 50 times more people than the Parkinson's disease, which is the fuel for today's industry. And you also need to understand that a subset of patients are candidates for surgeries, those who do not respond to medications, maybe 1 to 15% overall. And there is also a very slow and low adoption rate. In Cochlear, although it's very, very successful, it takes 10 years for people to really go from the time they're told they need a cochlear implant to the time they commit to it. You have this kind of external piece that puts a tag on on everyone wearing this kind of device. Parkinson's disease penetration is about 7% of all the clinical opportunity. Epilepsy is well below 1%. Retinal is below 0.01%, and others are pretty much zero because of the lack of better tools. So the conclusion is you can't treat use old tools to treat new indications, as already established in depression, Alzheimer's trials, which are ongoing, and so on. So there is insatiable need for better technology that can access functional networks of the human brain. So now imagine, also part of it is visualization, and this is developed in Professor Eddie Chang's lab at UCSF, this kind of visualization that you should be able to see through in 3 dimensions the structures of the brain, and imagine that you can now access multiple locations to stimulate on and look into the fibers for propagation and affected areas from which you record to truly manipulate functional networks of the human brain. Now, before we go there, we should also look into what technology concepts we can leverage to accelerate the development of this technology and bringing it closer to reality. So this is the Cochlear implant. It has battery-less implant device. It has magnet-aligned power transfer that transcutaneously transfers the power from the external battery-operated device. So this concept originated in 1957. It was FDA approved in 1985. Then there is the retinal implant, which is very limited in scope. It's only about 75 to 100 devices per year that are implanted. But perhaps this is the most challenging neurotechnology system, because you have electrode array that is attached to the retina, here in the middle. Then you have the RF coils for power transfer that is transferred through the transmit coil on the outside in this glass-mounted camera-based system. And you have to deal with the constant displacement of these coils with the eyeball movement. That is really challenging. And detachment of the electrode array from the retina, and then you have also another unit for the battery that is belt waist-mounted. So the problem with all of these technologies, with EEG caps, the DECA arm, the BCI, government, DARPA particularly, realized that these are technologies that are not clinically efficient or sustainable. And under the President's Brain Initiative, the big question was, how can we make these technologies more real, less invasive, beneficial to the patient, and sustainable and unobtrusive? And so that was the whole point of this Brain Initiative early on. One of the programs was the DARPA SUBNETS, in addition to the RAM, with the motivation to really address mental health issues in the military. And that included a whole range of indications, including PTSD, anxiety, depression, TBI, and a few others. And so here, the goal was to bring together device development, clinical neurology, and animal research to build a next-generation technology with more than 200 intervention contacts to really see what level of specificity is necessary for these next-generation indications, displacing existing devices with only 4 to 8 contacts that offer minimal benefit. So to answer to this question, we have developed entire de novo platform technology for research purposes. So there are 2 phases, phase 1 and phase 2, which correspond to generation 1 and generation 2 R&D. And so for clinical feasibility, there would be inevitable generation 3 translation. Nevertheless, this is a sketch of a system that's built under the DARPA SUBNETS project together with Dr. Eddie Chang from UCSF as the clinical PI. I was in charge of the technology. And so this included the implant system, which has control module implanted in the chest, aggregation module cranially mounted, and up to 4 cortical or subcortical smart leads with 64 contact interfaces. So this is for up to 256 channels with modern technologies for wireless power and data. with the external module that connects to the PC application and API that allows you to program the system. This technology was envisioned to be part of an end-to-end clinical system that would provide personalized treatment that is based on quantitative and objective assessment of electrophysiology in addition to behavior. So we had implantable device, closed-loop algorithm that would be individualized to each patient in addition to user technology for subjective mood tracking. Now, in order to develop technology for this, we built a platform for integration and miniaturization of all the electronics that always starts like this. So you have a whole bunch of printed circuit boards and circuits that you need to test. And typical, engineering PhD project is you build one board, test it, and you're done, have a PhD, publish a paper, and go out and graduate. So the requirement here was that you needed to build each one of these components in the context of the entire connected system. So we have the wireless power transfer on the upper left that is powering the stimulation and sensing boards, and in between we have a control module which runs embedded code that is programmed through computer that runs the whole system, and in this whole context the functionality of the system had to be demonstrated. Once you do that, then you can evolve these components to a fewer number of test points and higher level of integration. So we build this external device, which is a handheld device that you can take as an external trial device to verify the closed loop operation in actual clinical environment. And beyond that, you can further push the integration to an implant level electronics integration to be able to package this properly for an implant. So we have to go through all of these stages. And so this is the overview of the system. It senses activity from multiple brain locations, aggregates data to a hub, and sends it down to a chest module. In response, the chest module sends power and therapeutic stimulation that is individual to each patient. So the system has this smart lead, which is the world's smallest neuromodulator that is built to meet the human-grade requirements for sensing and stimulation. So you have a 64-contact vitamin pill-sized device that is attached to the lead that can be cortical or subcortical. You see that electronics here, it has several layers of chips that are integrated. So this is a sensor with a crystal oscillator, stimulation and power management with a medical grade capacitor, another stack of medical grade capacitors, and all of this was in the volume of 0.38 centimeter cubic to be modular and minimally invasive. Then the other piece is the control module, which is leveraging industry knowledge and packaging of our collaborators from Boston Scientific that provided us support to do the manufacturing and quality inspection of the electronics that was built to fit the mechanical constraints of the enclosure. These are the electrode arrays that are built that use 64 contact subcortical here and 64 contact cortical arrays that are attached to these smart leads. So this is increasing the level of resolution so that you can account for anatomical variations across patients and also find the correct contacts for each patient to individualized therapy. You don't always need all of the contacts, but you have the flexibility to choose the one that give you best benefit. So I'll give you now a bit of an overview from the technology perspective of the demonstration of some of these components and validation. So this is demonstration of concurrent sensing and stimulation, which was one of the key features we needed to provide. So this is the generation 1 of this vitamin pill 32 contact sensing and stimulation that is placed inside a smart lead device. So for this we had to develop a custom test board with a custom socket that can access these small features in a pogo pin fashion. So you have signal generation on the far right, power supplies, then the recording, and the software that is running on PC that runs the controls as well as puts the stimulation and displays the recordings. And this was done in saline here with electrodes to perform in vitro experiments. And so here is the reference electrode through which we inject the signal. And then on the actual electrode, we do stimulation and sensing. And so you want to see that you would be able to pick up this injected signal on the sensed electrode in presence of a very large stimulation artifact so that you can demonstrate full duplex sensing and stimulation. This is the user development version on which you have 4 stimulation engines. You can program the amplitude and also decide on which channel it maps. So you can see here 3.1 milliamps on this one that was programmed. You can also program the pulse width and display then the recording of stimulation plus artifacts. All this fuzziness that is riding along this waveform is actually from the stimulation artifact. We also realized that the world is not square pulses, so we built support for custom stimulation generation. You can put any kind of charge balanced stimulation waveform, and then you can program those amplitudes and change that from exponential to just about any shape. That allows you to put multiple frequency harmonics, to put exponential, which is more like natural brain behavior, and just experiment with anything that you can possibly think of. Then with that, we have developed initial considerations for adaptive stimulation artifact rejection that is based on 2 phases. The first phase is actually gathering responses at particular recording sites and using multiple recording sites to leverage mutual information for quicker convergence. And then you do the template detection and adaptive filtering to remove what you learned in the statistics. And basically this accounts for the fact that brain is nonlinear time varying system, that you need to do this adaptively. And once you apply that, you can plot signals in time and frequency domains, where you can see on the left without stimulation artifact and with stimulation artifact removal. You can see the clean signal in the time domain as well as the spectral domain. Now, in terms of speccing the range of your sensing electronics, you need to look into, oops, I think my PowerPoint crashed, so that happens. All right. So I think, where are we? We are down to oops. Okay. All right. So in order to determine what's the proper range for sensing electronics, we took actual experiments from human patients. So this is a right entorhinal cortex for recording of micro recordings from single unit recording in the presence of stimulation using Benke-Fried probe that I mentioned. So the bipolar stimulation is applied at this most distal end, and 7 millimeters apart from that, you're recording neural signal, and you want to see, as you ramp up the stimulation current, what is the level of amplitude that you need to have in order to do proper recording. And this is with understanding that this is recording performed with wall-plugged external instruments which have practically, infinite linearity for the purposes you need. But we want to do this at an implant scale, go from 40 watts to 4 microwatts if we can. And so that's the challenge. And when you see this signal amplitude with respect to the level of current from 0.2 milliamps, 0.4, 0.6, then you can see that you have, for this case, 20 millivolts extra dynamic range on top of your signal. So this artifact falls right inside your band. And so you have to provide this extra 5 bits of dynamics range without increasing your power consumption. And it's also important to note that these have to be linear bits because you do not want to have any unwanted distortion to your signal. And so we provided solutions for that. We leveraged actually significant infrastructure and training of students at UCLA in microelectronics in all aspects to be able to do all of these subsystems. And so we focused on providing support for very large input range. So we have 200 millivolts peak-to-peak sensing for both single unit and LFPs. And then very large input impedance, which is critically important here so that you reduce the number of off-chip capacitances. And you also need to tolerate large common mode signal swings because of the stimulation artifacts. So we provided support for all of that. And then, so these are all the pieces of technology. If you think about the whole system, then between UCSF and UCLA, we had to come up with the clinically driven system requirements and specs. And my group was in charge on system integration and all of the electronics. We have a whole bunch of vendors to provide electrode arrays, packaging by Lawrence Livermore, and then further chain of vendors to do these specialized tasks like ceramic PCB attachment, cables, and Boston Scientific for CM packaging. So there is a large number of people involved, and all of this had to kind of line up in time and specifications for it to work. This was DARPA D60 event exhibiting the technology and simple comparison with respect to current open loop technology and closed loop. You can see in open loop you're stimulating green all the time, and here you stimulate only in response to detected biomarkers. And we also had a patient video which was a big hit, which we had a patient with epilepsy, primary indication, who developed significant anxiety. You can find it on DARPA YouTube channel, and patient in presence of stimulation was able to claim, now I feel normal, which for a patient who has mental health disorder was a really significant breakthrough. And so this really kind of opens the doors to this kind of non-pharmacological treatment of mental health issues. In order to verify the technology, which is another whole stage of technology before the final validation, we had to build a system board that is envisioned to be able to test various components at different levels of maturity. You can see here the component that is put in a frame with a Goldfinger connector that slots into this interface. Then when that is successful in the context of the whole system, you excise that, you package it, and then you can have packaged component also verified on the same board. So we had the external module with computer, we had emulation as well as the hardware for the neuromodulator, we had the AM module, so the entire system components with full communication chain and control from PC application was demonstrated at government reviews for this technology. And just where we left off when we finished the program, that was at the end of phase 2 earlier this year, at the end of May, we came up with the entire system. I have some display toys here as well if you would like to see. So this is on top here is the entire implantable system. So this is pectorally mounted IPG and the control module with a data coil for real-time wireless telemetry. This goes through the neck into cranially mounted module, which is really just a data router and could be eliminated, and modular cortical and subcortical smart leads that are then attached to this module. So we did a full batch of production and verification of these components. So for example, here you can see a stack of all these printed circuit boards which are individually verified through a whole multi-chain inspection. We had 42 units that were sent over for packaging, and there is additional validation after packaging. Then we had external radio module here, which we built 20 of, and then we had functionality for both wireless as well as the wired connection through the UART cable. Then we had a PC application that runs the whole thing. And I think the two most significant things that I really find interesting from this whole exercise are that we were able to satisfy manufacturing requirements of the industrial level manufacturing by Boston Scientific to take electronics board that is entirely developed based on mechanical constraints and requirements for the operation, package that in a large quantity and pass 22-point inspection, which is like a 50-page report that you have to create for this to be able to say 37 of 47 units worked, meaning that they have passed all of the test points and were able to bring that into actual component that you can program the firmware and run on it from the PC application. And the other piece is this whole extreme level of miniaturization on the IP that relates to neuromodulator that I think we can take forward and refine for Generation 3 clinical technology. If you look into comparison of these technologies with respect to research technologies, which typically focus on electronics components only, then some science technologies such as these ones in the middle and therapeutic technologies, we were aiming to really be IDE compliant, to increase the number of electrode arrays, to provide low compliance, high level of stimulation with ±5 volts, to have large number of sensing channels and sensing capability that allows simultaneous sensing and stimulation, a radio capability that allows for real time, and implant device that is minimally invasive. Now, the invasiveness in this research demonstration wasn't really the key metric. It was more like leveraging existing components to bring this out to validation as soon as we can. But we inevitably need another iteration of this in which it's going to use much smaller number of components, meaning fewer points of failure and less risk, much smaller number of parts and vendors, and much smaller actually volume to be minimally invasive platform technology to support very large number of contacts. There is a number of references here that are with respect to these provide comparison table. And like I said, we envision taking this Generation 2 technology to Generation 3, which is going to look like something like that. Which is less than 4 cubic centimeters, that can be a platform for epilepsy monitoring. Like I said, it is a gateway for clinical translation of other indication-specific studies. Now, so there is a lot of things that need to happen, and we have a clear path how to do that effectively, not just for the electronics, but importantly for packaging and electrode arrays and connectors as well. Now you can also look into where are you with respect to the rest of the world and divide the neurotechnology landscape across the functionalities and various capabilities. So there is quite a bit that you actually need to take into account to understand what is the value that you're bringing out to the society. And so we've kind of gone through that exercise and feel that our solution is very unique and very timely. Before any kind of translation has to happen, you need to answer the question of clinical validation. And so with that in mind, we have been all along developing external trial device that I've shown to you, which I also have here with me, that is very quick to assemble. So we can scale this from 64 to 256 channels. That allows us to verify closed loop operation and all these features that people really desire, things like phase lock stimulation, full duplex sensing and stimulation, understanding the channel response. So we've done this, we cleared this through the clinical engineering at UCLA, we got approval for stimulation and sensing type of studies on human subjects, and we use this as a platform to inform Generation 3 implant design. Things like artifact rejection, you can only do that if you have brain in the loop and be able to finalize design specifications on that. In fact, we've done some recording already where the results are looking excellent. This is initial verification of sensing capability where we have done concordance analysis with neon codon system that you can see in the time and frequency domain they're practically indistinguishable, that we validate the quality of this device that we've made to match the existing clinical equipment. And the next patients will be doing also stimulation studies. We have fully automated kind of the exploration of these stimulation response maps, because one thing that I mentioned as you were looking through the features of the stimulator, the waveforms, the amplitudes, the shapes, and so on, that you really need to automate that. And so we've done it to the extent that we can access all of the channels, and with the clinician input we constrain which channels we can stimulate so that we can do complete stimulation response maps given the anatomical placement of the contacts to understand generally the behavior of the brain and all of these kind of response dynamics. I've also worked a little bit when I was on my sabbatical on another fun project, which was non-invasive technology, which is very successful. The concept here was to take EEG waveforms and translate that into computer music. So I worked with colleagues at Stanford, Dr. Parvizi and Professor Chris Chafe, to build neurotechnology that allows this to happen. And so we called this initially brain stethoscope. That was really like a stethoscope for the heart, that you can hear the sounds of the brain to determine whether a patient has epilepsy or not. And I'll just play you a few sounds to see how intuitive and simple this is. So this is a patient without seizure. What you hear is the background alpha activity around 10 hertz. Now here's another case without seizure. That's pretty simple, right? And so this is one brain with seizure. So here's another one. You kind of see this kind of pretty obvious, like these earthquake events. So we did actually give this survey to training, like 30-second training to high school students who at the end of the day had better seizure prediction accuracy than neurologists trained to look at waveforms. It was really nice to work with a neurologist who really wanted to lose his job to his technology, and technology is doing so well so far that everybody's kind of really looking forward to where this is going to go. This is a foundational technology for Cerebell, a very high-profile medical device startup. And so my conclusion here is that really this technology has the potential to revolutionize how we treat brain disorders. It can have implication on network scalopathies such as active memories and other indications. But even beyond medicine, I think interaction with these brain networks can really help us better understand the mechanisms of human memory and cognition and allow for treatment and augmentation of these functionalities such as assistive learning and accelerate human learning and knowledge. I would not venture to say anything about AI here because I think we really need to build our credibility and responsibility to the medical pathway before we can talk about going into AI issues with the human brain. So I think that will happen if we create deep-level intersects of medicine, technology, and neuroscience that would really help us accelerate the creation and adoption of these technologies in the real world. And my model as a researcher has been really in that direction. We need to create technologies for obviously training, publication, development purposes, but really having that practical aspect of how soon is this going to end up in a clinic and what are we going to get from it, right? So that's really kind of the end application and the purpose. And I like to say that, given the technology, the rate at which technology advances in consumer applications, I think we really need to take up our social responsibility and professional duty that in this day and age we should not be implanting telephone poles into people, but really kind of do something that's closer to the smartphone technology and capability that we have, and really seek ways in which we can collectively advance that to that maturity point that, again, will provide significant clinical benefit. So, thank you.
clarify the epilepsy neural stethoscope.
Yep.
Were those recordings for the patients with epilepsy during the baseline state or during an actual seizure? Which
On the right during the actual seizure, yeah.
okay.
And
what about their baseline? Is it any different?
Is it any different? That's a good question. Baseline is pretty much when there is no seizure, you pretty much see the baseline alpha activity. But when there is seizure, it's very easy to detect. And the whole idea here was that we wanted to kind of avoid the long pipeline of you see a primary doctor, you get a referral, you wait for kind of image scan, to have the technology that is really simple and intuitive that can be applied even outpatient. But we ran against a lot of issues like we originally thought naively this you can do with dry electrodes and like 2 electrodes, but there is really kind of requirement that mandates to have at least like 8 electrodes, that you have contacts on the occipital, both sides of temporal and frontal lobes to cover, and it have to be wet electrodes. So there is kind of a device which is a 2 kind of piece device. One is the display with all these like stethoscope sound functionality, as well as logging in all the raw EEG. And the other one is disposable headband that displaces gel to have the proper contact. So that's where it's.
Have you tried listening to any other kinds of brains with other conditions?
No, so far epilepsy, just yeah, only epilepsy.
So it sounds like about 40 Gen 2 devices have been created and exist on a shelf somewhere. And they're effectively IDE ready. So are there, sort of planned studies already under review and submission to implant them? And for what disorders, or is that, government secrets?
Yeah, so that's no secret. So I think that was the expectation, that we would be able to implant first in human 6 patients to verify the recording and stimulation with this technology. But there was no plan for a follow-on implantation study, right? So without which, obviously, we would not be able to proceed to do this. And I think, with resources, additional resources, that could be done. But it is also with understanding that Generation 3 technology would really have the right features to address the real kind of commercial and clinical need. The Generation 2 device, as you can see, it has what the FDA already recognizes and everybody is familiar with. It has a chest implant, chest piece. It has the depth or surface leads, but it also has this additional AM module. And so one way you can kind of position this is to say, well, if this device provides therapeutic benefit at the resolution and scale at which this was intended to work, then you can explant this system and replace it with the Gen 3 system, which would take a place of the AM. And then, still you need to work out the issues regarding the connectivity to the leads and things like that, which is non-trivial replacement. But I think really for this to be fully viable, you would need to get to the Gen 3, take the lessons learned from the R&D to reach the higher level of maturity to really be able to bring it out to the clinical. And that's the whole spirit of the external device, that as we develop this Gen 3 technology, all of the elements of Gen 3 technology are feeding into that external human study type of quality for recording and stimulation, and kind of establishing device-based record for future FDA submission in addition to the implant device itself.
So, but why create so many prototypes then? Why didn't you just make 5 Gen 2 devices? Why make 40 then, if you're not?
Yeah, so 40 was necessary for qualification. So if you look into the numbers you need for biocompatibility, sterility, and first in human use, So then you really kind of work through the numbers of what's necessary based on the volume of these devices. You needed to have a certain surface area, and that translates to a certain number of devices that you needed to do for biocomp sterility, packaging, and also human feasibility study builds that needed to be fully functional. So you kind of come up to the numbers that add up to 40 units. Out of those, we plan to have 6 systems for clinical. So basically 6 chest modules, 6 AMs, 24 NMs, and the rest was just biocomp sterility.
I was wondering if you could speak a little bit about the actual protocol for deployment in the clinic, because I think, if I'm understanding correctly, for this type of closed loop device, at some point you need to estimate a multiple input, multiple output model that's stored on the control module, and then I assume that that is lead localized. If you could talk a little bit about that process, first how you estimate those kernels, how they're deployed,
yeah,
whether or not they're localized, anything like that.
Yeah, it's a good question. So like I said, you have more contacts than what you need. So you first go through sensing kind of to figure out which contacts are relevant. And so out of 256 contacts, you will find that anywhere between 15 and 40, let's say typically 30 contacts are the ones that you care about. And you do that in multiple stages. You kind of do first system identification process. No stimulation, you kind of just see the baseline, and then you kind of figure out the biomarkers, and then you do the stimulation and make refinements. To the end, at the end of that system identification process, you then calculate which locations, which channels you need, okay? And then what you do offline as well, as you extract all of this data, you compute like various properties of those channels that are relevant and store them into kind of a matrix model for kind of running real time. And then aggregating all of those features into just like one metric, which is an algorithm. You can read papers by Mariam Shanechi from USC who developed the algorithm. But at the end of the day, it comes to one number tell all, which we call integrated mood score. Whatever that means, it's a one number that has a lower bound and upper bound. If it is below the lower bound, you involve stimulation. When it crosses the upper bound, you turn off the stimulation and kind of keep it adapting within those limits to decide when, where, and how to stimulate on 1 or 2 or 4 contacts in a multi-stimulation scenario. And so those matrices, once you define from the offline analysis, so for every patient, the protocol is you take all the data as you can, and it takes you a matter of days to aggregate enough information and say, okay, these channels matter, and now I'm going to compute these features. I'm going to program on the microcontroller, which was purposely built as a microcontroller to have that flexibility, and store this kind of model matrix on it, so that as the data comes in, you kind of take all those features and map them into a model that computes that one signal that navigates and triggers the stimulation. Now, stimulation you can do, again, fully adaptively, or you can jump between previously pre-configured and pre-programmed stimulation settings if you want to be more conservative. And then you kind of run stimulation in closed loop that way, and then you can do, again, periodic retraining and system identification every time you need to update stimulation parameters.